I thought, how difficult would it be? The protocol had been well established in cultured cells, and I just had to adapt it in the fly system. When I started doing the experiment, I thought it worked. 1 hour of enzyme treatment, and the polyphosphates showed up in 3 days. This was in November 2019. It was only in August 2020 that I realized the protocol worked a little differently in the fly systems. It took 9 months to realize this. Hello everyone, I am Shunarana Sarkar and I joined Manish Jaiswal's group in TIFR Hyderabad in 2019. When I joined the lab, Manish and I decided that I would establish the role of polyphosphates in Drosophila. This was quite ambitious because these chemicals have not been studied in the fly systems before. Naturally, this was also something that the lab had not worked on before. So I knew that I had an exciting but a huge responsibility to get this project off the ground. To all the graduate students who are pioneers in their lab-based projects, you would know that apart from being updated with the latest scientific literature to get a better understanding of your project, you also have to do a ton of reading to figure out which chemicals to use in your very first experiments. It's an additional task, and you're essentially creating an inventory of your own. So I joined the lab in June 2019, and it took 3 months to arrive at a list of essential items to start the experiments and order all the items off that list. So now to introduce you to this ambitious chemical called polyphosphates that all this preparation was for. When Arthur Kornberg was purifying enzymes from living systems to see what their functions were, he found out that there is an enzyme inside bacteria that could make long chains of only phosphates, and these long chains are called polyphosphates. We came to know of polyphosphate function in higher eukaryotes only very recently, about 20 years ago. And guess what? It is there to make your bones stronger, forming calcium phosphate clusters. When you get hurt, it is there to clot your blood by activating the clotting factors. It also acts as neurotransmitters and protein scaffolds. So you see, basically, it is almost everywhere. And for the past 20 years, people have been mostly working on in vitro systems, such as cultured cells, to discover these roles. But what we see in cells on a petri dish, is it actually what happens in a living organism? Or say, how does that change the behavior or functioning of the organism on the whole? That is what our lab decided to investigate. We are trying to address what polyphosphates do inside a living organism using fruit flies as a model system. The first step I had to take was to find out how much polyphosphates were present in these fly larvae. But then I thought, how many larvae should I start with? Would 50 larvae give me enough phosphates that the lab machines can detect? Or say, would 100 be a safer number to start with? I decided to go with 100. I counted 100 of these 3 millimeter long larvae and decided to extract polyphosphates from them. So why bother about how many larvae to take? Studies in the field have established protocols for cell cultures. Nobody has worked with flies to extract polyphosphates before. So it was very crucial to establish what is the minimum number of larvae required to produce enough polyphosphates that the machines could detect. Now, how do I extract polyphosphate from these fly larvae? What I did was crush the larvae and separate the long genomic DNA first. The extracted DNA looks like a white cloud floating in the vial. Now the solution apart from this cloud has the polyphosphates with a lot of other things from the cell. I was only interested in the polyphosphates. I took this juice and cleaned it up a bit by giving it a chloroform wash. After this, I added ethanol to separate the polyphosphate chains from the mixture. The separated polyphosphate chains show up as a small white pellet which can now be dissolved in deionized water. After that, what I had to see was the quantity of polyphosphate in this solution. For this, I divided the solution into two halves. To one portion, I added a phosphatase enzyme that would chop up the phosphate chains into single phosphates. And the second portion did not have any enzyme added to it, and it served as a control. Now, if you add a chemical called malachite green to solution, it binds to these single phosphates and forms a green colored compound. This helps us estimate how much phosphate was there in the larvae in the polyphosphate chains. There is a machine in the lab that helps us quantify the phosphates. Now, the machine gave me quite a good reading for 100 larvae. I was quite happy. The protocol worked. Collecting 100 larvae each time can be a so I needed to find out what is the minimum number of larvae which would give me enough phosphates to be quantified. I started with 5 larvae, no signal. Then I did the experiment with 10 larvae, still no signal. After this, I moved on to use 20 larvae, and finally, I got a signal. I could now do my experiments with 20 larvae. Mind you, it was already May and the pandemic was here. We had restrictions on and we could only come to the lab in given slots during the day. I got the 6:30 a.m. to 10:30 a.m. slot. Like any other graduate student, I overslept one day and missed the 6:30 a.m. shuttle to the institute. My experiment was already going on in the lab, and the phosphate chains would separate out into a pellet overnight. I asked my lab mate, who had not overslept like me, to dissolve the polyphosphate pellet in water and add the phosphatase enzyme, which would chop up the chains into monophosphates. I would take the next shuttle and resume the experiment. After adding the enzyme, the protocol in the literature said 1 hour of treatment for the enzyme to do its job. I managed to reach the lab that day 4 hours later. So the enzyme treatment had gone on for 3 more hours than what the protocol said. I was already skeptical about the success of this experiment. I decided to take the reading anyway. To my surprise, I saw there were more single phosphate molecules in solution from 20 larvae. Now this got me thinking. I went back to my lab book to look at my previous data. With 1 hour enzyme treatment, the amount of phosphates I was detecting with 20 larvae was same as 100 larvae. 100 larvae would of course have more phosphates in them, which means there was something missing in my protocol. Now, with the 4 hour enzyme treatment, I was seeing even more phosphate molecules from 20 larvae. This suggests a possibility that there are more polyphosphate chains from the larvae which need to be broken down, and 1 hour is not enough for the enzyme for chopping them all up. But all this while, I was not getting the entire picture of how much polyphosphate was there in the larvae. I was only skimming the surface. To straighten this out, I decided to do an experiment with different treatment times using 20 larvae. I had to standardize the treatment time. With no reference as to how much time is ideal, I decided to experiment with my own time points. You are supposed to have fun while doing science. I was having fun. So the durations for which I let the enzyme work were based on the shuttle timings during the COVID-19 pandemic. I let the enzyme work for the following durations: 2 hours, 5 hours, 16 hours, 18 hours, and 20 hours. Now, this was a shot in the dark. It could have been any time between, say, 5 to 16 hours, but I got lucky. 18 hours and 20 hours showed the same amount of single phosphates to me, which means that the enzyme needed about 18 hours to chop up all the phosphates that I have in my polyphosphate dissolved pellet. And it was August 2020 already. Remember, I started all this in November 2019. This was the story of how I standardized the protocol of detecting polyphosphates in flies. This was just the detection. This protocol that I established gave the lab a strong foundation to start addressing the bigger questions that we had in mind. And I learned I probably should miss shuttles more often.